Hello everyone, this is Justin, and today we will be talking about gene editing, CRISPR technology, and the recent news of Lulu and Nana. To start off, we first need to go through a quick biology lesson. We as living human beings are comprised up of different types of cells: blood cells, skin cells, muscle cells, etc. Each cell has their own job in the body, whether it be fighting off viruses, synthesizing new proteins, or carrying oxygen. The way cells know what their role is in the body is through DNA, a set of instructions that determine how our bodies function. DNA, or deoxyribose nucleic acid, is comprised of a sequence of smaller building blocks called nucleotides. There are four main nucleotides: adenine, thymine, guanine, and cytosine. Different combinations of these four letters can create different effects. This is called a gene, a distinct sequence of DNA that encodes function. For example, the TP53 gene is known as a tumor suppressor gene because that particular sequence of DNA codes for a protein that fights against cancer. On the other hand, the gene APOE provides important functions for cholesterol metabolism. We have thousands of genes in our bodies which determine the way we look, how our body operates in the world, and how it reacts to the environment. The genome is an organism's complete set of DNA, which means it contains all the necessary information needed to build that organism. So all your genes combined together make up the genome. By studying this, we can understand how our bodies function, our risks to diseases, our strengths and weaknesses, and even our ancestry. Companies like 23andMe and Ancestry.com have databases of people's DNA that can tell you your ancestry and even your health risks. After sending in DNA samples to one of these companies, they can tell you your risk for certain diseases like cancer. They do this by analyzing your DNA for specific genes. For example, the BRCA1 gene, when mutated, leads to increased risk of breast and ovarian cancer. By checking to see if you have this mutation, you can know whether or not you're at a higher risk for these cancers. This leads us to another discussion about gene editing. Gene editing is when you purposely insert, remove, or modify a specific genetic sequence of an organism. But why would people want to do that? Well, let's say you have a gene that puts you at risk for cancer. If you could remove that risk, would you? Alternatively, let's say we find a gene that correlates to extremely high intelligence. Would you want to insert that gene into yourself? There could be endless possibilities for gene editing and what we can improve upon in the human race. Currently, we have that ability to edit genomes. CRISPR technology is perhaps one of the most cutting-edge techniques created in the 21st century. Using the protein Cas9, which acts like a molecular scissor by cutting strands of DNA, scientists can purposely remove or insert DNA at their will. And while most of our discussion has been limited on the impact of gene editing to humans and disease, the implications of this technology are far-reaching even to agriculture and environmental ecosystems. For example, imagine inserting a drought-resistant gene into crops, which would allow consistent yields throughout the year. Or we could remove malaria-infected mosquitoes by preventing them from reproducing. However, this does raise ethical concerns. First, is it okay to play God? We could literally in the future control how people live, even how they are born. In fact, recently a Chinese researcher, He Jiankui, already created the first designer babies, called Lulu and Nana. Using CRISPR technology, they removed the CCR5 gene, which would normally allow HIV to enter the cells. In other words, they created two human babies who are inherently immune to HIV. And while some may think that this is a good thing, principally, perhaps we shouldn't be allowed to have that much control. Later on, maybe we only want people to have blue eyes or black hair. It's quite apocalyptic. But secondly, we also just don't know the consequences of gene editing. We've barely scratched the surface of how genes work together and how they impact each other. Removing one gene, like in Lulu and Nana's case, may have devastating consequences in the future and how they develop. And moreover, these consequences are not just limited to them, but also their children as well. They have essentially turned these two baby girls into guinea pigs, and perhaps even doomed their entire existence and future children. So what are the takeaways? Well, first, gene editing has both good and bad applications. It can help prevent disease and improve society, but it can also lead to grave ethical concerns when it comes to how we ought live our lives. And secondly, while genes can tell us a lot of information, our knowledge of how this works and how it impacts us is still very limited. More testing needs to be done before we even consider using it on humans, which is why there was so much backlash against the researcher. So what should you look out for? Well, look for breakthroughs in gene editing, perhaps new discoveries in genes that link to specific properties, or new ways to analyze the ramifications in changing genes. Thank you everyone for watching. If you liked this video, give it a like, subscribe for weekly content, and comment down below if you want to see me cover a specific topic.